Hello and welcome to our presentation on carotenoid research. To introduce you all to our research, I will break down what we did, why we did it, and how we did it. First is the what. Well, we tested and synthesized environmentally friendly methods of extracting provitamin carotenoids from sweet yams. Next is the why. One reason for conducting this research was for us to find an inexpensive way of studying these compounds. Another reason was for us to find a greener method of extraction. Lastly is the how. We used different organic solvents to extract carotenoids. We then analyzed the extracted mixtures using a spectrophotometer and thin layer chromatography to measure our results.
Now, you might ask yourself, what does this project have to do with global citizenship and gender equity? Well, first off, the research was conducted with the primary goal of finding viable green extraction methods. Our green methods of extraction of carotenoids can serve as a blueprint for researchers to decrease the amount of environmentally harmful waste from things like acetone and hexane, which is a not-so-green method of extraction that is normally used. Secondly, our research can potentially pave the way to gender equity through education. Replication of our research can serve as a learning experience to see the principles of organic chemistry in action. Women who face oppression in the education system can easily find the materials to do this kind of research, as the materials are only vegetable oils and yams. Our supervising professor will actually be doing half of our project for her first semester organic chemistry class as a laboratory experiment. And finally, gender inequity can be curtailed by combating malnutrition and increasing the quality of life in politically and economically disadvantaged regions of the world. The link between our research and malnutrition is our research works to help geneticists who are breeding vegetable plants to find a strain with the highest amount of provitamin carotenoids to help lower the rates of malnutrition, a terrible disease that, in some places, has twice the impact on women, according to the Food and Agriculture Organization of the United Nations.
The ultimate goal and purpose of this experiment is to know whether or not our environmentally green solvents, also known as our plant oils, can be used to extract carotenoids from our yam. And if so, how effective they are in comparison to our non-environmentally friendly solvents, hexane and acetone. Knowing this kind of information would mean that we have found an inexpensive and accessible way, as previously mentioned, way to conduct this experiment.
Implementation. The materials used for this experiment involves coconut oil, sunflower oil, grapeseed oil, and safflower oil. We also used sweet orange yams. The instruments used included a magic bullet blender, a benchmark scientific vortex, a Hamilton-Bell centrifuge, and a Spectronic 200. Sample preparation. A single yam was split between the class to use for each extraction. While the yam was washed and chopped, it was blended with 100 milliliters of DI water, and we used 4 grams of the slurry per sample. Method: extraction. We used 3 extractions. Extraction 1, we added 4 milliliters of solvent, vortexed 1 minute, and centrifuged for 2 minutes. We transferred the top layer to centrifuge 1 and labeled it. We extracted the bottom layer a second time by adding 4 milliliters of solvent, vortexing for 1 minute, and centrifuging for 2 minutes. Then we transferred the top layer and added to centrifuge tube 1. For extraction 3, we added 3 milliliters of water and a pea-sized amount of NaCl to centrifuge 1. We then vortexed for 1 minute and centrifuged for 1 minute. We removed the aqueous bottom layer and added a pea-size of magnesium sulfate to the organic layer. We vortexed for 1 minute and centrifuged for 1 minute. We collected the product by pipetting the solution into 25 milliliter volumetric flasks with microfilters, added solvent until 25 milliliter mark, and vortexed for 30 seconds. We transferred the solution into cuvettes and then measured the absorbance with a spectrometer at 450 nanometers and calculated the micrograms per gram of carotenoids. To set up a standard curve, we made dilutions of beta-carotene ordered from a laboratory and acetone as the solvent.
Data and results. Data collected includes mass of the yam, mass of the slurry, volume of the extract, and absorbance. Calculated data includes concentration of the carotenoids within the sample, final extraction value in micrograms of carotenoids per gram of sample. In order to calculate the concentration of samples, we had to create a set of standardized solutions with known concentrations of beta-carotene. Here we have a table displaying known concentrations of a standardized beta-carotene solution alongside their corresponding absorbance values. To the right is a calibration curve with absorbance on the x-axis and concentration on the y-axis. All of the oils except for the grapeseed oil performed similarly in terms of extraction value. The most consistent and desirable outcome was produced by coconut oil with an average extraction value of 206 micrograms per gram and a standard error of 8.24.
This table and the following table display more results from the grapeseed, safflower, and sunflower oil experiments.
In this slide, we have an equation used to convert the concentration value of our samples to our extraction value in micrograms per gram. In order to measure the probability that our data can be attributed to randomness, we performed a t-test that produced p-values for each oil compared to the conventional solvent. At 5% significance, our p-values indicate that our data is significant and support the idea that our data isn't a result of random sample error. This image displays our concentration values visually with bar graphs and corresponding error bars. Here it is evident that the coconut oil was far more consistent than any other solvent.
Discussion and conclusion. So as you can see from the charts from the data and results section, different oils extracted different amounts of carotenoids. Some possible explanations for this. One would be polarity. The polarity in the oils varies from oil to oil based on the, what the molecules inside the oil. So, as you may have learned in an introduction to chemistry class, like dissolves like. So if there's a polar solvent, it will dissolve polar carotenoids, for example, and extract those. But since there's a variance of polarity in the molecules in each oil, we are assuming that that played a big role in why some of the oils extracted more or less than the other oils. Another possible explanation is the carbon structure in the solvent. So going off of polarity again, some of the molecules in the solvents had bigger, smaller carbon structures based on the oil, and therefore played a role in the polarity, and which then again plays a role in which carotenoids it can extract, polar and non-polar. Another possible explanation is the viscosity of the solvents used. So the regular solvent, acetone and hexane mixtures are very non-viscous. They're very liquidy. They're easy to move around from container to container. Although with our oils, they are very viscous and they're very thick. So we hypothesized that some of the error was introduced when possibly when moving it from container to container. You'd measure out 4 milliliters or something, you only get 3.99, 8 or something like that. So we think that is where some of the error came from. The next possible explanation is photodegradation. Photodegradation is where objects are altered by light. In our case, this may have played a role because the carotenoids extracted from the yams may have deteriorated from the time that they were taken from the yam to the time that they were measured using a spectrophotometer, therefore increasing the amount of variance in our results. Finally, the coloration of oils played a role in the variance seen because, as mentioned before, photodegradation would lower the yields of the carotenoids taken from the yams, but it can also deteriorate the carotenoids from within the oils themselves, therefore offsetting the results to a degree. For future experiments, we could use charcoal to purify the green solvents and carotenoids to reduce the photodegeneration error. As I was just saying, in our solvents, we could take out the carotenoids in there, which do vary, especially in safflower oil. Therefore, we're taking away some of the variants introduced into our experiments. We could also use blends of oils based on their polarity. For example, we would take some polar oils and some non-polar oils and mix them together to optimize the number of carotenoids we could extract to a maximum, and that would probably provide to be more useful than just using one store-bought oil at a time. Another thing we could do is we could purchase light filters. Light filters could be purchased to put inside the lab to reduce the photodegeneration due to the lights. Some labs have these, ours did not. The next thing we could do is we could use a lab with the HPLC instrument. The HPLC instrument would help us to measure the amount of carotenoids extracted through high performance liquid chromatography, which is what HPLC stands for, but it's a very expensive instrument and we did not have access to one. The next two are kind of combined. They're separate methods of extracting carotenoids, not using toxic solvents, but those two in themselves we can go very far into them, but we will not in this presentation. Also, for future experiments, our research can be used in conjunction with interdisciplinary studies. Economics and sociology can be blended with chemistry to benefit people in politically and economically disadvantaged regions of the world. For future results, green solvents instead of popular toxic organic solvents can be used for learning purposes. Also, classrooms around the world, regardless of access to expensive materials, can benefit from our research.
We acknowledge Santa Monica College for providing the lab and materials. We are grateful for the STEM SRI program at Santa Monica College. We thank them for providing the Science 10 course materials and funding. A special thank you to Dr. Roman Ferede for supervising the lab, creating the experimental design, and teaching us new techniques and ideas. We acknowledge the stockroom staff at Santa Monica College for their help with creating solutions and buying materials. We offer a notable thank you to the lab technician Ronald Perez for preparing all of our samples and assisting us with the spectrophotometer. Lastly, we thank Dr. Mary Colavito, Dr. Amy Thompson, and Miss Vanon Yanian. Thank you so much, guys.